Yeah, welcome to our CBCS weekly seminar. We have this every week after morning tea with a variety of speakers from around the world and a variety of topics. CBCS is quite diverse and people are working across all the ecosystems. So you'll have a lot of different sort of people here with different interests. But I welcome you to our seminar. You are coming from, I'm just looking, you're coming from Stony Brook Uni. Where is that at?
That's on Long Island, about 2 hours east of New York City. Yeah.
Okay, welcome from New York. And you're going to talk to us about sensing in the dark, molecular ecology of bat senses. And I'll just turn it over to you. This is not my area, so I'm super interested to hear what you have to say.
Well, thank you, Carissa, and then thank you for everyone at the Center for Biodiversity and Conservation Science for inviting me to share some of my research with you. Before I get started, I want to also thank the National Science Foundation of the United States. They made possible the research that you're about to see. And you'll notice that I added a little asterisk to molecular ecology. And the reason for that is that there's more than molecular ecology. There's also going to be some morphology that we're going to look into today in today's presentation. And you're going to see a little bit of this logo with the bat, with the little bat flying towards the fruit. And that's the logo that we had for this project. This was a project we got a few years back with a bunch of wonderful colleagues. This is what's made this research possible. So at my lab, we actually have a double mission. There's more than one thing that we do. So on the one hand, in the blue, we tend to study these evolutionary processes that tend to increase biodiversity. And that's biodiversity, not just in terms of the number of species, but also in terms of traits or changes, changes that can open up new niches are particularly interesting for us in adaptive radiations. On the other hand, we have this other area of work where we are interested in the processes that tend to decrease biological diversity and that are happening right now around the world. And so this sort of at the center of everything is biodiversity. Biodiversity is the center of everything that we do. And these three areas that people in the background are students in my lab, but these three areas kind of define three large types of work that we do. In the blue, we've got questions about how biodiversity changes in time and space using phylogenetics, population genetics, those kinds of tools. In the center, we've got biological processes that fuel diversity, and that's got to do with genomics, taking a more functional approach to trying to figure out what the traits are doing and how they're emerging. And then on the red, we've got work that are factors, social factors and economic factors. These are coupled natural and human systems that are changing. Global change is evident around the world. What are these factors of environmental degradation, particularly in Latin America? However, it would take a long time to go through all of them. So today, I'm actually just going to focus on one of these, and I'm going to focus only in the biological processes that fuel biodiversity and using genomics and a little bit of morphology to examine that. As I mentioned earlier, this is not, I like to say that science is teamwork, right? It requires teamwork. And so these are the other three principal investigators on this project that we got from the National Science Foundation. Betsy Dumont, who's a morphologist in California, Karen Sears, who's at University of California in Los Angeles, and Stephen Rosser, who is at Queen Mary University in London. And so together with them, we just had complementary expertise and we were able to probe some of the questions that we're going to look into. So a lot of the time, if we're not familiar with bats, we're not necessarily familiar with all the diversity that they have. But looking at a picture like this, we suddenly see that there's a huge amount of diversity among them. We can see that the fur comes in different colors and their faces differ from one another. So we've got different sizes. If we're thinking about the sensory evolution of the bats, we tend to think about bats as all of them ecolocators or most of them ecolocators, and that's certainly the case. But you can see that sometimes we've got really massive ears and other times we've got tiny, tiny ears. Others, we've got eyes that are inside, eyes that are actually located pretty much inside the ear, the way that the entire face is taken up like a cone of sound. And we've got eyes. So a lot of people just kind of in part of folk culture believe that that bats can't see, but it's quite the opposite. They've actually got this diversity on eye sizes. Some of them have really big eyes, as you can see in this picture, and some of them have really tiny, tiny eyes. But all of bats can see. So this huge diversity is very intriguing because we know that bats are in this nocturnal niche, and we would think that this would lead to certain sensory specialization that would be different from other animals that are diurnal. But instead, we see this huge diversity and in nose leaves as well. And I want to draw your attention especially to this group that I'm highlighting here with the outline. This group is called noctilionoid bats. And noctilionoid bats are really fascinating because they include a diversity not just of sensory modes, but also a diversity of diets. So as part of this group of noctilionoid bats, we've got vampire bats, which are the only vertebrate group in the entire world that feeds exclusively on blood from other vertebrates. We've got bats that actually work like hummingbirds and they feed off of nectar in flowers, bats that eat figs, bats that catch fish, all of those, and bats that eat insects, which is what the majority of species of bats around the world do. So all of those are part of a single radiation that is mostly confined to the tropical forests and other ecosystems throughout tropical Americas. So on the islands of the Caribbean, and then pretty much from Arizona all the way to Argentina is where you're going to find the majority of this bats. So this is intriguing because this has happened in a relatively short span of time, that this huge amount of diversity in traits and in sensory biology has emerged. And some of the earliest work that was conducted trying to figure out in a phylogenetic context what was happening with this diversity of diets was published together with Betsy Dumont. At the time, we were very intrigued because we have this elongation of the face that you can see here. Some of the nectar feeding bats with the longest proportion of skull that you can see, and then at the same time, some blunt faces towards the bottom of this picture. You can see that this very blunt, short faces that are associated with the feeding on figs. And in this work, we discovered that this reorganization of the skull towards the blunt faces actually tracked the diversification of the group. And it was an innovation that enabled them to access this special niche of taking advantage of the figs of the tropical forests of Latin America. So that was really interesting, but it left out, well, what about all the sensory biology? And I was intrigued because, of course, one of the things that we see is that diversity in the eyes, in the size of the eyes and the size of the ears. And I was intrigued about the prospects for thinking about vision and olfaction in bats, which were understudied at the time. So today I'm going to focus on vision and olfaction, just these two senses. And I'm going to ask two questions, and I'll try to answer with some of the new data that we've generated for this project. So in terms of vision, does the feeding ecology that we just saw that's so interesting in the noctilionoid clade, does this feeding ecology actually shape the evolution of vision, as can be probed from the perspective of molecular ecology? And in terms of olfaction, does diversity in the diet of what they're eating, is it reflected also in the diversity, in the genetic diversity that we are finding in that part of the genome, which we will learn more about in a moment. So to get started, we're going to look into the feeding ecology and how it shapes evolution. So I want to add that this is what it looks like in Costa Rica, in La Selva, Costa Rica. And in order for us to do all the work that I'm presenting today, we had to go out into the field and we had to do this very elaborate collections, because only these elaborate collections enabled us to have the RNA, preserved RNA, that would enable us to probe into these transcriptomes from different organs in different lineages of bats. So that was actually quite fun to travel around in the forests of Costa Rica with a nitrogen tank. It's something that probably couldn't happen right now with all the restrictions. So getting back to this, right? So we're seeing that there's this division where we got these elongated muzzles that are associated with the feeding on nectar. And then we've got these blunt muzzles, and then we've got things like the blood feeding signal with the red, the vampire bats that are blood feeding, and a whole bunch of insectivorous bats. So looking at a framework like this in the phylogeny, well, we think, well, it's obvious. This is what's going to explain. We're going to find these adaptations, molecular adaptations, these genes that have been positively selected and have been changing under positive selection, and they're going to be associated with the diet. So what you're seeing at the top, what I'm highlighting at the top is all this diversity, how the different species that are shown at the bottom, with the phylogeny at the bottom, they have the different amounts of fruit, of nectar, leaves, fish and other vertebrates, or blood that they eat, right? So we have this tremendous diversity in their dietary ecology, and that matches the evolution twice. There's been 2 times that the evolution of feeding on nectar has happened in this group. There are several groups that are ancestrally insectivorous, and you can see the outgroups are all insectivorous, highly specialized in eating insects. And we were thinking, well, that's what's going to happen. The feeding ecology is going to explain the whole thing. So I'm going to take a little bit of time to explain this graph because it's trying to compile a lot of information. So you can think of this upset plot as a very elaborate series of Venn diagrams, right? So in the bar chart, we see the number of positively selected genes. And positively selected genes are these genes that are showing a signal at a particular node of the phylogeny is on the left. They are showing the signal of adaptation, right? So we're looking for these positively selected genes that perhaps are showing us that this particular function is being selected for. It's under positive selection and it's generating adaptations, right? The black dots and lines are a combination of positive selected genes that make up a cluster or a group. So for example, we see that there are 2 groups that are highlighted here in the orange, and those 2 groups correspond to the two nectar feeding lineages. So we see group F and G, both are nectar feeders, and they have 4 genes in common that are being positively selected in this sample of many thousands of genes that we were looking at. And then finally, the smaller bar chart that you see in the bottom left, it's the frequency count of the positively selected genes across the entire groups that you see. So knowing all of that, let's look. So what's happening is we've got this A, B, C, D, E, F, G, these nodes that are demarcated that corresponds to different diets. We were anticipating, remember, we were thinking that the feeding ecology was going to have a huge effect on the number of positively selected genes. Therefore, if that were true, we would find then that E and H and F and G would have the highest frequency of positively selected genes. But you can see right away that that's not the case. In fact, the vast majority of positively selected genes are actually shown all the way to the left. The vast majority are actually corresponding to the entire group, right? So we've got more than 400 positively selected genes in the ancestor of all the noctilinoids. And then the second highest number is in C. The ancestor of the phylostomids has more than 200 positively selected genes in this sample. So what is going on? Instead of having these huge numbers of positively selected genes that we were anticipating associated with the diet, we're instead seeing that the ancestor of the phylostomids and the ancestor of all the noctilinoids that we're studying in this sample have the most positively selected genes. And for example, when we were thinking, well, nectar feeding of flowers, there's something about flowers reflecting light in a particular way, even at night, that it would lead to a huge number of positively selected genes that would be shared by the 2 groups that feed off nectars. But instead we find that there's only 4. There's only 4 genes that are shared. So this was very puzzling and difficult to interpret. In fact, the paper was actually kind of difficult to write because we were still locked into this idea that we should have seen a lot more positively selected genes each time the diet changed. So it takes us in a completely different direction, and we need to have what I call a Batty aside. So Batty aside is that most bat species in the world, there's 1,400 bat species, more than 1,400 bat species, and most of them are aerial insectivores. So we're going to see this brief video of what that looks like. What does it look like to be an aerial insectivore? And that's exactly what's going on here. There's the moth, and then the bat approaches the moth, and there's a lot of behavioral work, a lot of acoustic work, detailing how bats actually are listening in to the sounds that the moths are making, and they're using their echolocation, and then it uses its tail and the skin in between its legs called the patagium. It's using this uropatagium to grab the moth and bring it to its face, and it's eating that. So the vast majority of bats are doing something like this when they're catching insects. But there's something very peculiar that happens with the noctilionoids, right? Noctilionoids, including all the species that I've just been showing you, they are not aerial insectivores. Instead of being aerial insectivores, noctilionoids are gleaners. What does it mean to be a gleaner? Well, this is the fishing bat Noctilio. Noctilio leporinus is the fishing bat. And so what it's doing is actually catching its prey off of a surface. In this case, the fishing bat has been shown to actually use echolocation as well to actually look for ripples in the surface of the water. But once it senses a ripple, it actually goes and catches it off the surface of the water. So that's what we call gleaners. And this is very different. This foraging ecology it differs from all the other aerial insectivores that we were just looking at in the previous one. So what kind of genes are popping up? So we went to the field, we caught all these bats, we got all these tissues, and then we examined these genes that were expressed in the eyes, that were expressed as mRNA in the eyes of different species of bats with different ecologies. So what does it look like at the different nodes? Well, remember that the vast majority are actually being found in the ancestor of all the noctilionoids that we have in our sample, which are all neotropical, or in the ancestor of phylostomids. So a lot of the genes, if we actually classify them into gene ontologies or into the functionality, what they're doing in the ancestor of all noctilionoids, we got homeostasis of the retina, responses to salt stress, and associated in the ancestor of nectar feedings, we've got detection of stimulus, lens fiber, cell differentiation, and in the ancestor of the phylostomids, we've got response to salt stress. And this is actually kind of really interesting because, of course, we know that one of the things that happens in controlling the eye, the blood pressure in the eye, is that this is actually a very difficult thing to control, that in us humans, as we grow older, it becomes more difficult, right? And so that's one of the things that we need to be watching out for in the process of aging. And what we're seeing is that in these ancestral bats, ancestral noctilionoid and ancestral phylostomid bats, there's actually a lot of positive selection associated with responses to salt stress and with some kind of hyperosmotic response to dealing with that, associated with that. Of course, this is all done from homology, and we don't know that that's exactly what these genes are doing in the bats, but it provides us some hints as to where to look for these functions. So when we evaluate and we find out that this ancestor of all noctilionoids in the sample and phylostomids are the ones that are changing, well, one of the things that became clear is that these changes towards positive selection in the genes that we were finding were not associated with the individual diets, with the feeding ecology, but instead were associated with this shift that happened in the ancestor of phylostomids, but also in the ancestor of noctilionoids towards foraging using gleaning. And so the ecological basis of this, or the ecological association that we were looking for originally just with the diet, is instead happening with the foraging. That's fascinating for reasons that we're going to see later in the talk, but just enough to say that this gleaning, the same gleaning that enables the fishing bat to catch that fish off of the surface of the water, the same gleaning is an approach that these bats, descendant of phylostomids bats, they use the same gleaning to catch insects off of leaves. So there's this long-standing hypothesis that doesn't have any, that hasn't received a lot of support, but there's a long-standing hypothesis that this contact with the gleaning, this catching insects off of leaves, expose them to this very different diet, and that this may have changed their physiology forever. So that's interesting. All right. So in positive selection, in these eye-related genes that are expressed in the eyes, we thought the adaptations were going to be related to the feeding ecology, but instead the adaptations were related to the foraging ecology, and especially to the gleaning foraging ecology. This molecular adaptations trace back to the evolution of gleaning rather than to the evolution of specific diets. The specific diets are associated with some adaptation, but it's really minor compared to what happened. And so here's a picture of a bat from the Caribbean, a brachyphylla bat. You can see that it's completely covered in pollen. They're nectarivorous bats. Nectarivorous and frugivorous bats, and you can see right there that it's involved in pollination. So far, I've been talking about genes, thinking about molecular ecology, talking about this, only genetics. But what about morphology? Could the actual form, right, follow the function? And could it also match this model of adaptation associated with foraging instead of adaptation associated with feeding ecology? So here's one way to find out. And of course, this is part of a collaboration. I forgot to mention that the previous work and paper was published by Kalina Davis, who's a postdoc in Stephen Rossiter's lab in London. And so this is Ronnie Hall. He's a graduate student at University of California, Merced. And Ronnie Hall wanted to quantify the sensory systems of the bat. So what did he want to do? He actually wanted to separate the olfactory bulb, the orbits of the eye, and the cochlea of the bats and actually study them. And this is only made possible. So we're all used to looking at CT scans and looking at the bones using the CT scans. But in the last few years, a series of innovators, including a former student of mine, Laurel Yo, have come up with methods to actually look into specimens that are conserved in collections and examine them and look at the soft tissues. So instead of just looking at the bones and instead of just quantifying the bones, we can actually, as you can see in the images at the top, you can see muscles. Sometimes you can see, like you can actually see the encephalus, you can see the brain, and you can see the orbits that are present in that CT scan. And it's thanks to this iodine staining that is being used. So once you've done this reconstruction, so in the case of the cochlea, of course, it's bony, you can get the bony cochlea, but you can actually reconstruct parts like the olfactory bulb as well. And this paper was highlighted in Evolution this year. And so Martinez and Naas did this really fantastic illustration here that I highly recommend that is associated with our paper. It's a commentary on the findings. And their point is that we're trying to look at, so our starting point is that the bats, unlike other mammals, they're very limited. There's very limited space. So you actually expect that there's going to be some correlations happening, and in some cases trade-offs, because you cannot grow the head very large, because you cannot make a bat very large. You cannot make a bat very large. There's a ceiling on the size that you can achieve as a bat because of light. And because of that, then we expect to be able to relate one thing to another, to relate the size of the orbit to the size of the, we're expecting trade-offs. So we're expecting negative relationships between the sizes of the orbit, for example, and the olfactory bulbs and the cochlea. So what did we find out after doing this, all of this CT scanning from hundreds of specimens? Well, what Ronnie found out is that definitely after we correct for the size of the specimen, because remember these bats come in different sizes, the eyes and the olfactory bulbs are actually positively correlated instead of negative. So we had predicted that there was that these are sort of competing for space somehow. But instead we have this roughly this positive relationship. And the cochlea, when we're looking at the cochlea are kind of on their own. We don't really have a correlation with them. They're just kind of changing on their own. The cochlea are doing their own thing. So what does that look like? How does this all fit into the head of the bats? Well, once more, we've got the outgroups over on the left, so the insect outgroups, and then all of the ones that you see in colors at the bottom, vertebrates, omnivore, nectar, and figs, these are phylostomid bats. So they are part of our in-group leaf-nosed bats of the neotropics. And these leaf-nosed bats, we can see right away that compared to the outgroup, there's something different about them, about the real state that's being occupied in the head. There's a gigantic amount proportionally that is being given over to olfaction. And the cochlea have already become smaller in that ancestor of all the leaf-nosed bats. They're smaller. But we can see that certain diets also change. So the cochlea are smaller, and then they become really small in some of these derived nectar feeding and fig eating groups. But also the fig eating bats have the largest eyes. Proportionally, they end up having really large eyes compared to this, compared to the samples. And then definitely compared to the outgroups just have this really gigantic cochlea. The cochlea is just massive compared to them. So the question is, well, we have this kind of very rough comparison that's kind of binning them into this foraging ecologies once more. But when did this happen? When in the evolution of did this bats change so much from that ancestral plan in which you have large cochleas and small olfactory bulbs to this plan in which you end up having smaller cochleas and large olfactory bulbs that we see here? And so that's what we're seeing here. I've taken out some of the more extraneous and left the same color scheme. And I want you to draw your attention towards the bottom rows of Phylostomid and the ancestor of Noctilinoids. Because we see right now that Phylostomidae, which is the new, the leaf nose bats of the neotropics, already have the ancestor, if we do this ancestral inference, already has a larger olfactory bulb. So that ancestor had already shifted. That shift had already happened early on. And that ancestor Phylostomid, as we can see on the top right, already had a larger orbits. So the eyes became larger and the olfactory bulbs had become larger already. Proportionally, the cochlea had become smaller, though not significantly smaller. As you can see, there are error bars around that. So we can see that it is on average smaller. It's a size adjusted smaller cochlea, but it's not significantly smaller than the other bats. And then finally, there is something interesting happening with the nectar feeding bats, that they have the smallest cochlea of them all. They end up having tiny, tiny cochleas that are tiny and significantly smaller than everything else. So to answer the question, when did this all happen? Again, it happened ancestrally. It didn't happen, this kind of new organization of the head in which we have bats with small cochleas and larger olfactory bulbs and larger eyes is happening in the ancestor of the Phylostomids as a whole. And there are some shifts that happen later, like for example, nectar feeding bats evolving tiny cochlea, but we don't see the same degree of reorganization happening. So once more, we're finding this pattern where the ancestor of the Phylostomids is already has evolved this presumed adaptations or this sensory changes that are correlated with a diversity of diets that seems to have appeared much later. So to sort of wrap up this part about looking at the morphology, well, we expected once more that the shifts would be related to diet, but in fact, instead we found that this omnivorous sensory morphotype was ancestral. And what I mean by omnivorous sensory morphotype is a sensory morphotype that included large olfactory bulbs that may be associated with being able to actually recognize fruit, and we're going to talk about that in a little bit, and also larger eyes that may also be associated with this finding of fruit. So the question that's puzzling us, and this is a question that we post now in 3 different papers, 2 with molecular data and 1 that is with morphological data, is are we looking at pre-adaptation? Is it the case that these bats somehow had acquired all of these traits and then their diets changed and they were able to make use of these sensory systems, even though their ancestor was still feeding off primarily of insects, like the vast majority of bats? Or is it the case that when they switched their foraging and they started catching things off of leaves, these bats just accidentally started eating a whole bunch of leaves and started eating a whole bunch of fruit? Well, not a whole bunch, tiny amounts of fruit, tiny amounts. And so this ancestor of the leaf nose bats actually started being able to process and started evolving this complete difference. And I don't have an answer for you yet. We've written 2 papers where we've actually talked about pre-adaptation. We've talked about this ancestral omnivorous morphotype. But here's an interesting bit of data. This is not an ancestral, of all of the leaf nose bats, of all the Phylostomids. This is a fossil reconstructed from 12 million years ago in the Inter-Andean Valley of Colombia. And this place called La Venta holds some of the oldest remains that are definitely demonstrating an association between plants and bats. What do I mean by that? This tooth, which we see on the top right, that tooth is distinctly associated with nectarivory. This bat, because of that tooth, And the bat is actually known only from 2 teeth that kind of look like that. That bat ate nectar. It included nectar in its diet. But in 2015, with my former student, Laurel Yo, we decided to look more carefully into, first of all, where did that bat belong in the phylogeny? And what was the body size of that bat? And was that consistent? What was the size of the skull for that bat? And was that consistent with exclusive nectarivory and with the proportions of the head? And what we discovered when we actually kind of looked at all the nectarivorous bats in this radiation is that it was consistent with a mixed omnivorous diet. So we've got this 12 million year old bat that was already omnivorous. What do we mean by omnivorous? That the bodies, the head size, the inferred head size of this bat, keeping up the proportion of nectarivorous bats that are alive today. The head size is consistent not with exclusive nectarivory, but it's consistent with a mixed diet in which you have nectar, but insects are also being included. And this is a really fascinating reconstruction. Every animal that you see here, the hoatzin, the crocodile, the marsupial, all the animals that you're seeing here have been found in the 12 million year old beds of the Magdalena River of Colombia, where this fossil is found. And so what we did is, using the combination of the phylogenetics and the reconstruction that we had made with our regressions, we actually did this reconstruction of what that bat would have looked like, right? The phylogenetics told us that it would have been one of these orange bats that exist, or similar to these orange bats that are nectar feeding today in the forests of South America. And the size, the reconstruction of the size told us that it would have been relatively large, and its skull was wide enough to indicate that it wasn't super specialized. So we think that what we're seeing is that perhaps instead of calling it pre-adaptation, we should be thinking about that ancestor of the phylostomids as being omnivorous, as having this diet that already went down in the trophic level and included some plants. And then all the descendants, all the different branches that descend from it include some kind of plant matter in their diet at some level. But of course, there's a lot more to do. All right, so the next question is, with the sense of olfaction, the sense of smell, and the question there is, does diversity beget diversity, right? So the sense of smell is, of course, one of the most fascinating ones. It's fascinating because, you know, like it is extremely difficult for humans to actually communicate a sense of smell to another person. So when we try to communicate that, it's hard to verbalize. And most of the time we're just saying, can you smell that? Can you smell this? And of course, the sense of smell has become front and center now that there's this COVID pandemic. And one of the things that happens is that the sense of smell changes for people, right? That there is a significant proportion of the population that has had COVID that actually doesn't fully recover their sense of smell. So I'm going to walk through how the sense, you know, some of the foundations of this, so that we can get us to what we're doing with the bats in terms of all of this perception. So the perception of smell begins with receptors, right? This is a model of a mammalian olfactory receptors that's embedded in a biological membrane. And olfactory receptors are, of course, a very diverse group of genes that we're going to see. So the model, the best supported model for how this happens is that there are these volatile compounds that are floating around, and they bind to this specific receptors, right? There's the specificity. And after binding, the receptors activate a switch that begins the mental representation of the odor going towards the olfactory bulb, and from there to other areas of the brain communicating, talking about that. So in terms of genetics, so just, you know, let's forget the neuro for now and just focus on the genetics of this, right? The people start dropping around. Okay, so I'm seeing some things. All right, so in terms of genetics, olfactory receptors are actually fascinating. When they were discovered in the 1990s, the person who discovered them, Linda Buck, she actually got the Nobel Prize in 2004. And the reason why she got the Nobel Prize is that we didn't understand the molecular biology of them. And olfactory receptors end up being the single largest protein coding family in the entire genome, right? So in your entire genome, there are a whole bunch of genes that are protein coding, and the largest group of them, between 3 and 5% of them in any mammal, a mouse, a rat, a dog, a cat, is going to be olfactory receptors. It's the single largest group. And so what we have is this vision that we have this source, for example, a mouse or a cat, and we have this odorant, this volatile odorants that come off of that, and they match like a lock and key. They match each other. And therefore, if I'm a mammal that wants to, that actually requires as part of its daily life perceiving this big diversity, for example, another cat or, or another mouse or a piece of fish or a hamburger, then I'm going to keep these receptors. These receptors are actually going to be part of my genome. That's how we see it. And this happens through a process of duplication and gene loss as well. But gene duplication is actually super hard to study because it requires very high quality genomes to know where, side by side, because they're highly similar. They just duplicate. Instead of just evolving a little change, they actually just duplicate in whole, the olfactory receptors. And getting high quality genome is expensive. Right now, it costs about $20,000. So even the analysis, the models within and between species are still not quite settled. So what we came up in my lab was we came up with an affordable solution. So we went from having to spend, bless you, we went from having to spend $20,000 or $30,000 doing a very good genome to actually using sequence capture, which you see at the top, which is about 90% as good as getting the genome. But instead of costing $20,000, it costs something like $200. So we generated this kind of innovation in how we can get the olfactory receptors from non-model organisms. And we wanted to answer the question, does diversity beget diversity? So are we going to see positive selection and adaptation? And we were doing this with 3 species of bats, of leaf-nosed bats of the neotropics called Carolia. And these 3 species have a stepwise increase in the number of pepper species that they eat, of pepper specialization. So Carolia castanea is the smallest of the 3, and it feeds almost exclusively on pepper species of plant. Carolia sowelli is a medium intermediate size bat at La Selva in Costa Rica, and about half its diet, or a little over half of its diet, is just made up of pepper plants. And Carolia perspicillata is the largest of them, and it actually includes lots of things in its diet, including insects and a lot of other plants. So using this approach of the bait capture for capturing the genes and sequencing them at very low cost, we were able to actually scan the entire diversity of olfactory receptors of these 3 species, as you can see here. So we can see the 3 species, and we can see that there are different subfamilies of olfactory receptors that are present in each. So what we wanted to find out is, well, is it the case that there's more diversity in olfactory receptors in the one that is more of a generalist, that has a generalist diet? And is it the case that the one that is very specialized has fewer? So we used a phylogenetic approach in which we looked at these clusters, and we measure the diversity using the Shannon-Wiener index, which you see here in plot A. And indeed, we find that on the whole, there's less diversity. The diversity index for the species that's highly specialized in the dark green is actually lower than for the species that's more diversified, which is shown in the yellow. So we find an indication that using this phylogenetic approach and then measuring the diversity, that indeed diversity does beget diversity in this case. And this is what it looks like. This is one of the small Carolia bats. And this is actually not a pepper plant, but this is how they eat. They actually just kind of eat like corn on the cob. Follow. So we were thinking, well, molecular diversity is related to dietary diversity. And in this case, indeed, that's the case. Is there positive selection and adaptation? It's actually really hard. And I'm sure that there are people in this audience. One of the premises of studying in molecular ecology, studying positive selection and adaptation, is that we're actually looking at orthologous genes. We're actually looking at the same copy in every species and at the same copy in every individual. But of course, with this process of duplication, that makes it very confusing. It's extremely difficult to figure out which one is the orthologous copy and which one is a paralogous copy. So that makes it hard to look for molecular adaptation. It's complicated by this process of duplication and loss, which is something that several labs are working on and which we definitely have the data for. So I want to just quickly, it looks like I've got the right time. Okay, so does feeding ecology, I wanted to answer two questions. Does feeding ecology shape evolution? And does diversity beget diversity in the molecular ecology of the senses? Well, what we find when we study vision, both the molecular ecology and the morphology, is that foraging is the factor that has really changed adaptations. This visual sensory adaptation, and this kind of more complex proportions of the olfactory bulb, the cochlea, and the eyes are related to foraging instead of diet transition. When we look at olfactory receptors, diversity does seem to beget diversity, but we still don't know. We still don't have the toolkit or the models that are advanced enough to take in this much data and to account for the fact that we don't actually have orthologous genes every time to talk about adaptation very clearly yet. And with that, I want to thank you. And I want to, of course, thank all the members of my lab for making this work possible. Thank you.